So, TikoMed is our main company. We have a subsidiary called TikoMed AB, and where we actually do the work on this development. TikoMed was founded by me in 2002. I wanted to get into this this booming industry of cell therapies that was going to cure everything and you know, lower your tax and get your car running on fumes. It didn't happen in 2002. I think we're all aware of that part. But I did discover a couple of scientists who had done a breakthrough innovation in islet cell transplantation, where they discovered a reaction called IBsolvMIR, instant blood-mediated inflammatory reaction. The effects of this IBsolvMIR reaction is essentially developed by Uppsala University in Sweden, a couple of researchers there called Olle Korsgren and Bo Nilsson. And they could see that cells, or the islets, were killed off by this innate immune reaction quite instantaneously after hitting the bloodstream when they were presented, and supposed to travel through the portal vein to the liver and graft there. They went through a number of different compounds to find one that would alleviate this reaction, and they found one that we were working on that's called IBsolvMIR. In 2004, we went through heaven and hell. We got funded by the NIH. It was a bit of a special journey because we got that funding in 2004, and we had the first patient in 2009, and received the final report from NIH in 2017. And, well, probably the slowest clinical study ever. So we had to do some other things, and hence the neurology leg on that. But now it's 2018, and we have received some results, and they're very encouraging. So therefore, I'm here to further this. On the way, we managed to get orphan drug designation for islet cell transplantation. We are continuing with studies, and we have managed to file a number of new patents. This is our board. Beautiful, aren't they? And this is my management team. We are not so many in that team, but we have a lot of consultants. So this is the IBsolvMIR reaction. It starts when the cells or the islets hit the blood. Well, essentially the islets express tissue factor and activates a thrombotic reaction, but it also activates a lot of inflammatory cells, etc., that just makes the blood clot around the cells and eventually kill up to 70% of the graft. Well, it says 50 here, but that's been documented in several rounds. The first discovery was, of course, in the islets of Langerhans, because that's what they were focusing on at the time. And we can show now that this reaction can be alleviated by our drug. So instead of losing about these 70% of the graft, we seem to save about 70% of the graft. And surrounding this IBsolvMIR reaction, there has been a lot of international independent publications. So we think we have a very, very interesting compound here, not only for the diabetic patients, but also for patients receiving other kinds of transplantations of various cells. So the molecule is a low molecular weight dextran sulfate. It's infused together. It's given as a bolus dose just before the cells are infused. I will emphasize that low molecular weight dextran sulfate is an old molecule, but they really do not look the same. They are very, very different in character, and we have developed this one to be one that really fulfills what we think is important to alleviate this reaction. What we have documented over the years now is that this drug has benefits just before even the cells enter the system. It has effects during the actual transplantation and then after. So what we have seen is that we can improve the in vitro viability of cells. They will come to a study on that. We have done several more than the one that's going to be displayed, but that one is quite revealing. So as I mentioned, it's an efficient inhibitor of IBMIR and actually the whole sort of process. We have shown also that we reduce lung entrapment of cells, which I know is very valid for mesenchymal stem cell transplantation. We have anticoagulant factors, and we stimulate the stem cells to get more active. And post the actual transplant, we have shown that we can stimulate growth factors to improve the engraftment. So, we have done quite a lot of tox studies involving both rodents and non-human primates. We have been able to go very high in dose, giving us a vast margin on dosing. And we have also done quite an extensive safety program. So this drug has now been dosed to 79 healthy volunteers and a bunch of, 10 patients. And doses between 3 and 24 milligrams per kilo have been very well accepted. We have had no significant adverse events and no SAEs. So we are now planning for follow-up studies. Here is just some data showing that with various sorts of cells, we have repeated this in various conditions. This one is quite interesting because this is a study on autologous cells for patients with pancreatitis, chronic pancreatitis, where you could resect the pancreas, isolate the islets, and transplant them back. And the interesting thing for us here is that autologous cells also elicit this IBMIR reaction. And we have shown this part about engraftment of the hematopoietic stem cells. So we have done different sorts of cells. We have done various situations. And, this is probably the strongest publication for us with IBMIR, showing the direct reduction of IBMIR. And here is the lung entrapment part, showing that T cells transport better through the lung together with this compound than they would do on their own. And we see a lot of that with entrapment of T cells getting into the fibrous tissues of lungs. Let's see. And same thing here. We increase the survival for engrafted myoblasts. So again, yet another cell type, yet another good result. And this is the specific stuff we have done with the survival of islets. This was sort of the foundation that we published together with Professor Masafumi Goto. It was a really good eye-opener for us. And we also looked at liver cells. Well, we have done specific studies with mesenchymal cells, stromal cells, etc. And with the same outcome, same result. We help them survive prior to transplantation. We help the transplantation process in turn and in the end, we help the cells engraft. So next step here is to run a clinical program in islet transplantation. We will do it at 2 sites in Sweden at this point. We are planning to expand it to other European countries. We're going to do cadaveric donors at this point with 18 patients receiving 1 transplantation each. Treatment will consist of 1 bolus dose and then do 3 follow-up doses over a week post-transplantation to enhance the engraftment process. So Karolinska and Uppsala will be the first, the universities there will be the first centers that we work with, and then follow up. The next planned study for us will be in hematopoietic stem cell transplantation for MS patients, the ones that have an early onset, but also a fast progression. It's been a very successful treatment in Uppsala, and we aim to add to that process on those patients. So we are here to partner. We think we have a drug that's shown great safety, good efficacy. We are now looking for companies that would be interested in benefiting in a collaboration, companies that are in clinical phase, about to enter a clinical phase 2, and wants to add this drug onto the program to enhance the survival and reduce the graft loss in their therapy. We have a vast patent portfolio. Actually, we have just added a couple of patents to this program and these patent families. There's a number of publications coming out, and these are actually both our own publications, but also publications made by external investigators who have looked at, done independent study. And I will thank you for listening.